All right, so a quick review before we carry on of all that we've covered so far. Uh, we started with chapter 3A because we are following the syllabus pattern for Cambridge. So that's cell structure, and the things we learned are microscopy, which is the use of the light and electron microscopes for how to prepare specimen, the magnification resolution. We're talking about the internal structures of the cell and the ultrastructure, organelles such as nucleus and mitochondria, protein secretion from cells, and the organization of cells into tissues and then organs. Now, cells are the basic unit of life, and they are the fundamental unit of structure, function, organization, and living organisms. They are very small. The largest cell known is the human X cell, which is the only cell that's visible by the naked eye. You can naked cell over here, it's a bit embarrassing, by the naked eye. We just correct that. Okay. Then moving on to the light microscope. It is a small and portable device. It is inexpensive, so it's easy to have at hospitals and even universities and schools. Basically, that's where it really counts.
Yeah,
Maybe the more advanced hospitals and universities can afford the electron microscope, perhaps, But the light microscope is something that's available to almost anyone who needs it. It can magnify something up to 1500 times. The X over here means times in magnification, And that means how large an image is compared to its actual size. The formula for magnification is image size over actual size, and we can move this about to find out either the magnification or the image size or the actual size, depending on what the unknown is. So we make the unknown the subject of the formula and move everything about, so you can get three different right, setups for that. For the light microscope, the specimen is prepared by straining. It is put on a slide, and then the stage of the light microscope. There's a light source underneath the specimen, so it's very crude drawing over here. There's a light source, right? And the specimen somewhere over here. There's an objective lens. There's the first level of magnification, and then the light travels up to an eyepiece lens, and that's supposed to be an eye over there for the observation. And so there are two lenses through which magnification occurs, and so that's a multiplication effect. So if the objective lens has a magnification of times 10, and the eyepiece lens has a magnification of 15, so you get 150. There are various stains available. Some examples are methylene blue and acetocarmine, and they are used to stain different types of cells and different parts of the cells, So if you want to stain a specific organelle, for example, uh, and they give different characteristic colors. The advantages, as mentioned, they are small, they're inexpensive, they're portable, relatively cheap, so inexpensive, and living tissue can possibly be viewed under the light microscope. Disadvantages, you can end up with artifacts, and that is common for the light and electron microscopes. So the structures that appear due to the preparation and staining process that are not cellular structures. You see something under the microscope, and that's not actually, and the magnification is relatively low, So just about 1500. Coming to the electron microscope, there's a lot of processing to prepare specimen, such as dehydration, freeze fracturing, and you need a vacuum inside the electron microscope, so that they don't interfere with the electrons that are the source of radiation. Uh, right, living tissue cannot be used. The electron microscope is useful because it can give a magnification up to 500,000 times. right? So electron beam is passed through the specimen, and then right electromagnetic lenses, and they are focused onto a screen where the image is produced. And it is because of the electron microscope that the fine structure of the cell surface membrane, the nucleus, right? So these are things that are visible by through the light microscope, but right, they're more detailed structures of these organelles, and then other organelles uh, were also made visible due to the electron microscope, and those are called the ultrastructures. Example: resolution of a microscope is right how clearly it can tell two different objects apart, right, when they're close. So the eye can tell apart two objects when they're as close as 0.1 millimeter, right? Closer than that, and you can't make them out. The light microscope can tell apart two objects that are as close as 0.2 micrometer. The electron microscope has a high resolution. It can tell objects apart that are as close as zero point 0.1 to 1 nanometer. Right, so you can easily distinguish between two objects. So vesicles fusing with membranes, or proteins attaching to receptors, or however whatever is going on in the cell. And that's because electrons behave as particles and waves. So that's the wave particle duality, not exactly part of the syllabus, that's from physics, but that is the concept behind it. And so they have a high frequency, right? And so they can actually pass through that narrow space, that really small space of 1 nanometer between two objects, and resolve them as two separate objects. Yes,
Are you sharing your screen?
just a minute. All right, you can see it now.
Yes.
Sorry about that.
Questions.
Okay. All right, so a micrograph is a picture obtained from a microscope. The transmission electron microscope, micrograph, gives you a 2D image, right? It has a lower resolution. Sorry, it has a high resolution, but it's a 2D image. Scanning electron micrograph gives you a 3D image, but it is comparatively low in resolution as compared to the 2D transmission electron micrograph. Sometimes colors are added digitally, and so those are false scanning electron micrographs. The advantage of the electron microscope is the magnification and the resolution, obviously. The disadvantages are that you cannot study living material. There's a lot of preparation involved, and so a lot of artifacts can show up. It is expensive, not accessible to everyone, and it needs to be maintained at a constant temperature and pressure. So, a lot of care is required. Then we move on to eukaryotic cells. Right, so common cell structures are what we'll be talking about, and common is what you'd expect a cell to have. It's not necessarily that all cells do have them, or how many of them they have, or what if there's any specific difference in an organelle depending on the function of the cell. Right, they mention eukaryotic cells because right, eukaryotic cells are one that have a proper nucleus as compared to a prokaryote, right, which do not have a nucleus. They do have genetic material, but right, pro means before, and karyote means nucleus. U means true. So, right, prokaryotic cells don't have a nucleus. So we're talking about the eukaryotic ones at the moment. Common structures, right, are the cytoplasm, right. So there's fluid inside the cells. You have the cell surface membrane. You have the nucleus, right. Maybe you can see a few ribosomes, and sometimes, as I mentioned, the structures may be missing in some cells. So red blood cells do not have a nucleus. The quantity may vary. So mitochondria, right, how many of them are present depends on how active the cell is. So cell surface membrane is a double layer surrounding the cell. It controls the entry and exit of materials, and also keeps the contents of the cell contained. The nucleus is the largest organelle in the cell. It contains genetic material, the DNA and RNA. It's a dense, there's a separate dense mass called the nucleolus, which is responsible for the production of the ribosomes in the cell, right? The nucleus is also surrounded by a double membrane or a nuclear membrane, and there are pores for materials to enter and leave, called nuclear pores. The cytoplasm is a jelly-like material in the cell and contains all the organelles and other stuff, chemicals, proteins, hormones, maybe, right? So everything else that's going on in there. And all of this combined is called the protoplasm, right? So all the living material of the cell. When you move to the ultrastructure, as I mentioned earlier, it is a right, and that was obtained using the electron microscope. So that shows the details of the organelles and structures inside the cell, right? So, right, something I was observed that there are a lot of membranes inside the cell. So there's obviously the plasma or cell membrane. There's a nuclear membrane. There's a membrane around the Golgi apparatus, the endoplasmic reticulum. The mitochondria has a double membrane. Lysosomes are membrane-bound vesicles, right? Vacuoles, right? And right, so these membranes can be, right, intracellular or extracellular. Then more detail about the nucleus. It is 1 to 20 micrometers in diameter. It controls the functioning of the cell because it has a DNA which transcribes and translates into proteins. Right, the nucleic acids are DNA and RNA, along with proteins that help them coil up or stabilize them. When when the cell is not dividing, the DNA is bound to proteins to form chromatin, right? So that's a certain level of foiling, right? And as I mentioned, the nucleolus has DNA and protein as responsible for the production of ribosomes. The mitochondria, and we've done this in detail when we talk about respiration, they're rod-like. They used to be called the powerhouse of the cell because it is here where ATP is produced through aerobic respiration. They are 1 micrometer wide and 10 micrometers long. They have a double membrane, and depends on the activity of the cell how many of these are present. So there are very few of them in fat cells, a lot more in muscles and liver cells, thousands per cell. They have an outer membrane that surrounds the organelle and an inner mitochondrial membrane, which is very much folded, right? And that is called, the folds are called cristae or crista, and that increases the available surface area for respiration. And lastly, the point where we had reached, right, the fluid in the mitochondria is called the matrix. They have their own DNA, and mitochondria replicate independently when the cell is undergoing division. And this points towards the endosymbiotic theory, where it was believed that the mitochondria and the chloroplast were single-celled organisms, eubacteria. They were engulfed by eukaryotic cells and ended up with a symbiotic relationship where both the mitochondria and the eukaryotic cell were benefiting from each other, and that is how it has been ever since. Okay, and then finally a learning tip: when you're mentioning the structure of any cell or organ or tissue or organelle, always try to relate it to the function. Right, so if there's a lot of infolding in the inner mitochondrial membrane, you mentioned the function that increases the surface area for respiration, right? For the reactions that take place that produce ATP. That is a review of what we've done so far. Any questions?
No, sir.
All right. The next organelle that we're going to talk about are the centrioles. And they play an important part during cell division, right? So there's usually a pair of centrioles near the nucleus, right? And they are made up of, it's each centriole is a bundle of 9 sets of microtubules, right? So there's a pair, and each of those is a bundle of 9 sets, actually triplets of microtubules. Right, and they're about 0.5 micrometer long and 0.2 micrometer wide. Right, so as I mentioned, they're involved in cell division, and they're actually responsible for pulling the replicated sets of DNA apart, right, to each daughter cell. Right, they achieve, so obviously we get more detail about this later, because they produce a spindle that goes and attaches to a certain point of that replicated DNA and pulls it apart. Next, we have the ribosomes. Right, and this is where protein synthesis. This is the organelle where protein synthesis occurs. Right, so the DNA, when it is read and RNA is produced, so DNA, right, a particular gene is read and it produces RNA or mRNA to be specific, right, and that moves out of the nucleus and goes to the ribosome, where it is translated, right, into an amino acid sequence, and those amino acids are joined together by peptide bonds to form proteins, right, or actually to be more specific, right, polypeptides. I'm not saying proteins directly, because a single protein can have one polypeptide chain. It can have more than one polypeptide chain, right? So it depends on how many of those it has. So the amino acids are joined together to form polypeptides, right? So these macromolecules. The RNA, the ribosome, right, is made up of ribosomal RNA. Right, and the organelle has a large and small subunit. Now, when you're talking about that, there are two types of ribosomes in eukaryotic cells. Right, there is the ATS ribosome, which is present in the cytoplasm, right? So freely, it's present freely in the cytoplasm. It's also attached to the endoplasmic reticulum, right? Uh, the rough endoplasmic reticulum to be specific, right, where protein synthesis occurs. And right, the S stands for the Seidberg, right, unit. So that's basically a sedimentation. Let's make this a capital S. Right, so the S, right, stands for Seidberg. Right, which is how fast, right, it settles to the bottom, or how far down the tube it goes during centrifuge, when it's put into a centrifuge. So, right, ATS means it's a heavier ribosome, and so it will sink lower when put into a centrifuge. And right, so you could kind of use an analogy for mass, right? That AT is like the mass. It doesn't actually work that way because the large and small subunits, right, have their own Seidberg units. So the ATS ribosome, right, in that the small subunit is 40S and the large subunit is 60S. And so you would assume that if these two combine, the ribosome will be 100S, but that's not how it works. Right, so when these two subunits combine, they make the ATS, and so that's how quickly and how far down the tube they go when put in a centrifuge. There is another ribosome called the 70S ribosome, and this is the one that's present in mitochondria. Because remember, it has its own DNA, and so it produces its own proteins as well. Right, so it has its own ribosome, and also in chloroplasts, because chloroplasts also fall under that endosymbiotic theory that they were eubacteria once, and then the plant cell took it up. Excuse me. Right, and these, right, they're also the ribosomes found in prokaryotic cells, and again that you know leads back to the endosymbiotic theory for mitochondria and chloroplasts. They also obviously have the large and small subunit. So the small subunit is 50S in this case. Sorry, 30S. That's for the large one. The small subunit is 30S, and the large subunit is 50S. And you would expect, oh, this is going to be the one that is 18, or this one is 7. Right, so it's not the simple math. Okay. And one thing they've mentioned is, right, if I just go back, that in the ribosome, the ratio in the ATS ribosome ratio of RNA to protein. Right, so the ribosome also has certain proteins along with the RNA to give it its structure, is 1:1, right? Whereas in the 70S, the ratio for RNA to protein is 2:1. So there's more RNA to the protein that's part of the organelle. Any questions so far?
No, sir.
Okay. There's a lot of information coming in, right? So
Yeah.
yeah. So lysosomes. Lysosomes are membrane bound vesicles containing digestive enzymes. Right? So, right, these are organelles that are present in single-celled organisms such as the amoeba, where they release their enzymes to digest food in their environment and then take it up by diffusion. Right? Then they're also present in our body cells. Right? And the name lysosome is derived from lysis, right, which means breakdown. starts breaking down. Now, when observed in a cell, they appear as dark spherical bodies in the cytoplasm. Right, the digestive enzymes that they have, so the lysosomes can fuse with the cell surface membrane or the plasma membrane or the cell membrane and release the enzymes outside the cell. This can happen in the gut where they're releasing the enzymes to digest protein. So as we know, the digestive enzymes, it can also happen if the cells from which lysosomes are being released, so they could be white blood cells, and they are targeting foreign bodies, pathogens, bacteria, something that's got into the body that shouldn't be there. So they release the digestive enzymes and digest the bacterial cell, or old, damaged, dead cells, body cells, those are also digested this way. And what lysosomes do, the digestive enzymes, one of the digestive enzymes present in the lysosomes is called lysozyme. Not surprising. So that's a very generic thing. And what these enzymes do is that they break up whatever molecule they're acting on, like whatever substance they're acting on, into molecules. So they break up the substance into molecules, which are reused. So unless it's a pathogen which needs to be gotten rid of, if it's a cell or something else, obviously in the case of digestion, it's broken down into molecules which can be taken up and used or reused, whatever the case is, and so not to waste resources. Lysosomes can also self-destruct. So that's an interesting thing, and that is called apoptosis. Or programmed cell death. And that is when the cell becomes too old or damaged, or there's a mutation that's occurred, and that could possibly lead to it becoming cancerous or harmful in some way to the body, and so the lysosomes inside the cell they rupture and release their digestive enzymes, and end up destroying the cell and everything in it, and so that is sort of self-destruct, as they mention in the book. And something that's given in the book additional information is there obviously anything can go wrong in the body, and that's why there's so many different types of diseases and so many variations of those diseases. And And right, so something can go wrong with apoptosis as well, right? That lysosomes begin self-destructing cells that shouldn't be undergoing cell death, right? So they should be okay, but something goes wrong because of which programmed cell death occurs, and that right would end up in disease, right? Because it is not controlled, it is not what should be happening in that particular scenario. All right, are you still with me?
Yes, sir.
Okay. Next up, we are going to talk about endoplasmic reticulum. These are fun names, aren't obviously they're not new. Right, the endoplasmic reticulum. Now we know there are two types of them. There is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Right, so it is a network of membranes. In general, if you're talking about these in general, right, it's a network of membranes in the cell, and it's not just localized; it can be spread out throughout the cell, right? And so these membranes appear as sac-like cavities or tubular, right, structures. So they can be sac-like cavities or tubular structures. And they're spread throughout the cytoplasm, as I mentioned. The right ER, we call it, right, is linked to the membrane of the nucleus. Whenever you see an ultrastructure, you will see that right the endoplasmic reticulum is closely attached to the nuclear membrane. Now, what is it involved in? It is involved in transport and protein synthesis. In the cell. They say that about 1 centimeter cube, right, of liver tissue has about 11 meters square of endoplasmic reticulum. Right, so that's a lot. This is the volume. So a volume of 1 centimeter cube of liver tissue has an extensive network of the ER, which gives a surface area of 11 meters square, which is massive. Right, now obviously it is folded and cramped and thin, and so that's why. Right, so this is just the surface area, right? Obviously these are on a microscopic level. Okay, so there are two types of endoplasmic reticulum. We have the rough endoplasmic reticulum, the rough ER, and we have the smooth endoplasmic reticulum, or right. You can also call this the RER, and this is where right there are ribosomes attached, and right protein synthesis occurs. And so the protein is synthesized, and on the ribosome, and then it is put, transferred into the ER, right, or the rough the reason the rough endoplasmic reticulum is called the rough endoplasmic reticulum is because it's a network of membranes, and the rough part are the attached ribosomes. So if the polypeptide chain is synthesized on the ribosomes, and then they are transferred inside the ER, right? So inside the membranes, where they are modified, right, and they achieve their structures. So the tertiary or quaternary structure, which will come to later, and then they are transported, right, in vesicles. So cells, right, that secrete substances, right, chemicals or substances, just to be more general, right, have a more extensive network of you know rough endoplasmic reticulum. And so the more active they are, the more endoplasmic, the rough endoplasmic reticulum that you are going to have. And right, if they are, if these are proteins that are going to work outside the cell, then they are put into vesicles, moved to the cell surface membrane, and then moved outside. For example, we talked about lysosomes, right? So they also secrete digestive enzymes to the outside, right? And doing that is called exocytosis. And so you want whatever substance it is to not interfere with the cell, right? Just like you want it for the lysosome as well, right? Because then when it's released inside the cell, right, it causes cell death. And so whatever it is that is being transported, right, from the rough endoplasmic reticulum is taken to, right, I mean, it goes through the Golgi apparatus as well, so we'll talk about that. But right, it fuses with the membrane outside, it is compartmentalized, and that is called exocytosis. Then we have the smooth, right, and this is the last thing I'll be talking about today, smooth endoplasmic reticulum, or the SER, right, and these are ones that do not have ribosomes. So they're not involved in protein synthesis, but they are involved in synthesis and transport. That is right for steroids and lipids. So remember that ribosomes are specific to protein synthesis, but proteins aren't the only thing that are going about in the cell. So you have got steroids and lipids. So cells which are secreting chemical substances made of lipids, made of steroids, right, will have more active smooth endoplasmic reticulums. For example, right, cells in the male reproductive organ, right, the testes, which release the steroid hormone testosterone. Right, so that's a steroid hormone, and so that is synthesized and then transported from the smooth endoplasmic reticulum. Sure. You also have a lot of the SERs in the liver, which metabolize cholesterol and other lipids. Cholesterol and lipids. And so finally, the type and the amount of the endoplasmic reticulum in a cell gives an idea of what the cell does, right? It's a secretory cell. What is it secreting? The type of chemical substance that it's secreting. Is it a protein, a lipid, a steroid, right? And yeah, that's it. So next, which will be next week's class, unfortunately, I guess. All right, we'll move on and talk about the Golgi apparatus, which is, I mean, it's actually the last of the organelles that we will talk about before moving on to prokaryotic cells. Right? So that's just a bit left, unfortunately, right? We're out of time for today. Any questions?
No, sir.
That was a lot, wasn't it?
Yes.
All right, so I do recommend you go and read the book. All right, so that's chapter 3A2 and 3A3. Right, from the
Yes.
Edexcel textbook for AS. So just write them down. The Edexcel single book 1 for the AS chapters 3A2 and 3A3. And you can obviously look at 3A1 as well for revision if you want to. I did go over it at the beginning of class. And yeah, okay. So I'll see you tomorrow for A2 inshallah.
Inshallah.
All right then. See you, Mami.
Hafiz.